السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. Dear students, I am Dr. Rahma Kamaluddin Abu Noor, Professor of Histology, Faculty of Medicine, Cairo University. I'm going to give you Cytology, the first chapter in Histology. First of all, we should introduce what the meaning of histology. The word histo, tissue, ology, science. So it is the science that deal with the tissues. These tissues should be normal in structure, and I should use microscope, because if they are not normal, we are dealing with another subject called pathology, and if I'm not using microscope, we are dealing with anatomy. So it is the science dealing with microscopic structure of the normal components of human, animal, and plant, formed of cells. The cells collect to form tissues, the tissues collect together to form organs, okay, and the organs form the system of the body. Histology should correlate the structure with function. Cells should be examined under the microscope. Why? Because the cells are minute in size, ranging from 4 to 150 micrometer. So it must be examined under the microscope. You should know the meaning of magnification power and resolution power. The word magnification power is the degree of enlargement, while resolution power is the least distance between 2 points. You can see them as 2. Resolution power, you can see the slide clear. You should know units of measurements, that 1 centimeter contains 10 millimeter, 1 millimeter contains 1000 micrometer, 1 micrometer contains 1000 nanometer, and 1 nanometer containing 10 angstroms. We said that I should use microscopes. What are the common used microscopes in histology? We have 2 types of microscopes, light microscope, and the initials is LM, and electron microscopy, and the initials is EM. The common used, as we said, the LM and EM. This is the LM light microscope. What I can see here, this is the eyepiece, this is the tube, and these are the objective lens. Here we can put the slide on the stage, and this is the source of illumination, either electricity or the sun. And here is the gross focus, and this is the fine focus of adjustment, and this is the arm from which we hold the light microscope. This is an electron microscope. It's very huge and should be present in a certain room. How to use the electron microscope? It works with electron beams, and we can watch the slide using a special stain called osmic acid at a very high magnification. What is the difference between light microscope and electron microscope? First of all, the source of illumination of the light microscope depends on the daylight or electric light, while the EM beams of electron. Stain used, we use many stains in the light microscope, and this is and can give us many colors, while the electron microscope I have only one stain, which is called the osmic acid. If it stains something positive, it appears black. If it does not stain, it appears white. So we are dealing with shades of gray. The magnification power by the light microscope 1000 or 1500, and the resolution power is 0.2 micrometer. While the EM I can magnify more than 100,000, and the resolution will be 0.2 nanometer. The field examined by the light microscope, I can see many cells, by the EM, I can see one or two cell or even part of one cell. Okay, let us examine ourselves. This is light or electron microscopic picture. This is light. Why? Number one, I can see many cells. Number two, they are stained red and blue, or pink and blue. Number three, I cannot see details of the cell. I just see nucleus and the cell membrane and the cytoplasm. I cannot see details. Okay, look here. This is electron microscope. Why? I can see only one cell. Two, I can see details. Three, it appears gray. Okay. This is scanning electron microscope. We are dealing with the surface of the structure. This type of photo, you're not going to see it in our study. When I see a slide under the microscope, it should be stained. The most common histological stain by the light microscope is called hematoxylin and eosine. H stand for hematoxylin, E stand for eosine. From its name, this stain is formed of two structures or two chemical components. Why? In order to see two different nature of components. One of them will stain acidic component while the other stain basic component. The hematoxylin stain any acidic structure in the cell. Any acidic structure in the cell. The most common acidic structure in the cell is the nucleus. It contains nucleic acid. So this acid will be stained with basic stain and will be called basophilic and appears blue as it is stained with hematoxylin. Okay. The eosine stain anything basic in nature. So the basic in nature will be stained with acidic stain and will be called acidophilic. And the stain is red and it's called eosine. So examine yourself. If this slide LM or EM, it is LM. Why? I can see colors, many cells, no details. So, and this stain is hematoxylin and eosine. Why? All the nucleus appears blue and the cytoplasm either blue or red according to the reaction inside the cell. Okay. Cytology is the science that deals with the cell, and this is the most important chapter in order you're going to build up more information about histology. Why I'm studying the cell, the first chapter? Because the cell is the functional and the structural unit of all living tissues. It is the structural and the functional unit of all living tissues. Cells have different shapes and size. The cell is made up of nucleus and the cytoplasm. I never call a cell by this name unless it contains nucleus. Is there any part of our tissues that does not contain nucleus? Yes, the red blood corpuscles and the platelets. Okay, so we are going to deal with cells that contain nucleus and cytoplasm. Nucleus and cytoplasm. The cytoplasm is formed of matrix, organelles, and inclusions. First, I'm going to study the organelle. The meaning of the word organelle, small organ. They are living structures. They are doing chemical, containing enzymes, and they are vital, producing essential function in the cell. They are differentiated. Each organelle can do a specific function, and they are permanent, usually present in the cell. I cannot live without them. Inclusions, the difference of organelles. They are not essential for the vitality of the cell, maybe stored food. Some of the inclusions are very important as the myoglobin and the hemoglobin. They are oxygen binding proteins. These are important inclusions. The matrix between the organelles, nucleus, and the inclusions, there is a matrix which appears as colloidal structure formed of protein, carbohydrates, enzymes, minerals, irons. Okay, so this is the nucleus and this is the cytoplasm. The cytoplasm contains organelles which are differentiated, permanent, essential, and the inclusions which temporary, usually not vital, and we have the matrix. The organelles, we have either membranous organelles surrounded by a membrane, as the cell membrane, mitochondria, endoplasmic reticulum, rough and smooth, Golgi apparatus, lysosomes, peroxisomes, non-membranous, does not surrounded by a membrane, as ribosomes and cytoskeleton, as well as proteasomes. This is diagrammatic picture. You should differentiate between diagrammatic LM and EM picture. First, we're going to study the cell membrane, or the other name, plasma lemma. This photo is EM or LM. It's EM. So when I say to you cell membrane and I put a picture EM, this means that it's very fine and it's difficult to be seen by the light microscope. The cell membrane is very thin, surrounding the cell. It's very difficult to be seen by the light microscope. If you want to see it by light microscope, you should stain it with silver or a stain called PAS. By EM, it appears as two dark lines separated by a light one. This is called trilamellar, formed of 3 layers, plus fuzzy layer found on the outer surface only, which is the cell coat. Again, look here, dear students, this is a cell and this is another cell. And here is the cell membrane surrounding this cell. I can see that is formed of 2 dark lines separated by 1 pale line, and this is called trilamellar. I can see 3 lines. On the outer surface, I can see something called fuzzy layer, which is the cell coat, and I'm going to explain it for you now. Again, this is one cell and this is another cell. Here is the intercellular space containing the fuzzy layer, as it is the outer part of the cell. In order to understand why it's formed by trilamellar, you should understand the molecular structure of the cell membrane. It's formed of lipid components, which is phospholipid molecules and cholesterol. The phospholipid arranged in 2 layers. Each molecule is formed of head and tail. The heads are charged directed outwards toward the aqueous solution at the inner and the outer surface of the cell. The hydrophobic, or the tails, they are non-charged and they are directed toward the inside of the cell, facing each other. While the cholesterol molecule present between the tails of the phospholipid, and the function is to limit movement of the phospholipid, make it more stable. Here is the phospholipid bilayer. The heads are directed outwards and the tails directed inwards. Between the tail, I can see the cholesterol molecule. Again, this is a diagrammatic picture. I can see the phospholipid bilayer and the tails directed inwards. Okay, from this I can explain why I can see 3 lines, 2 dark and 1 pale. The heads were going to be stained with the osmic acid and appears dark. The tails were not going to be stained, so it appears pale. And the heads again will be stained with the osmic acid and appear dark. Okay, the second component of the cell membrane is the protein. We have either extrinsic protein, they are small and non-continuous, they are attached loosely on both surfaces of the cell. Intrinsic protein, either they are small molecules or large passing through the membrane called transmembrane protein, and they form channels for the water-soluble substances that cannot pass through the phospholipid layer. Again, here is the phospholipid bilayer. Here is the tails directed inwards. Here is the protein, either peripheral or intrinsic protein. Then the carbohydrates formed of a sugar chain. The carbohydrates either combine with the lipids forming glycolipid or combines with the protein forming glycoprotein, and this is a mistake in the diagram. It is glycoprotein. The carbohydrate components usually present on the external surface only. Why? First, I'm going to tell you the carbohydrates fused with the protein forming glycoprotein, fused with the lipids forming glycolipids. Both of them form something called cell coat or glycocalyx. Why? It's present on the external surface only because it contains receptors. And the receptors should be present to the outside of the cell in order to combine with different drugs, hormones, bacteria, or virus. The carbohydrate components, as we said, is usually conjugated either with proteins or lipids, forming the surface coat. This surface coat of the glycocalyx is responsible for the fuzzy appearance of the cell membrane by the electron microscope. Again, this is the lipid bilayer. This is the carbohydrates forming glycolipid, and here is the carbohydrates fusing with the protein forming glycoprotein. This is the transmembrane proteins, and this is the peripheral protein. What is the function of the cell membrane? The main function of the cell membrane is transport. We have many types of transport. The first is passive transport. What the meaning of passive? It does not need energy. It moves with concentration gradient from high concentration to low concentration. Further, we're going to divide the passive transport into simple, in which the gases or water moves with concentration gradient along the phospholipid layer, while facilitated diffusion, it needs a carrier, which substances that are not soluble in the fat move by concentration gradient through the transmembrane protein, integral protein, which acts as a carrier as the passage of glucose and amino acids. What the meaning of active transport is transport of molecule against concentration gradients, and this needs energy. The best example is sodium pump. All the time I said to the sodium, go out of the cell. Okay, so the concentration of the sodium will be more outside of the cell than inside. So it will enter the cell by what? By passive. And I said to it, you should go outside. Moving the sodium from inside to outside needs energy because outside is more. Okay? Look here, dear student, this is the passive transport and this is the active transport. Why this is a passive? Because it moves from high concentration to low concentration. Why this is positive? Because or active? because it moves from low concentration to high concentration. What is the difference between both the upper and the second one? This moves through the phospholipid bilayer, so it is passive simple. This moves again in the transmembrane protein, so it's called passive facilitated. And this moves against concentration gradient through the transmembrane protein, and this needs energy. Okay? Again, the sodium outside is more than the inside, and usually I'm saying to the sodium, go out. So it moves against concentration gradient. The third type of transport is called bulk transport. By its name, it's something big. It's going to be dissolved and pass through something. So what I need, I want to enter a bulky structure inside the cell. Entering such structure is called endocytosis, to the inside of the cell. It includes phagocytosis, I'm eating something, for example, bacteria or food. Pinocytosis, I'm entering fluid. Receptor-mediated endocytosis, I selectively entered something to the cell. This photo, something outside the cell wants to enter. I'm going to make something called pseudopodia. Pseudo means false podia pedicles, and the structure will enter taking part of the cell with it. I'm going to deal with it, and after dealing with it, I'm going to do something called exocytosis outside. While I'm doing exocytosis, the cell membrane that entered during endocytosis will be left inside the cell. Okay, so there will be no change in the integrity of the cell membrane. Endocytosis. Here is the cell membrane. This is the solid structure they want to enter. I will send pseudopodia, false food, okay, that will engulf the solid particles, fuse, entering part of the cell membrane together with the something that I want to eat. This is called phagosome or food vacuole. So, what's the meaning of phagocytosis? Cell eating. I'm engulfing solid particles, bacteria, dead cell, foreign body, food, forming phagosome or phagocytic vesicle that pass inside the cytoplasm. Pinocytosis. This is the cell membrane. It will invaginate and the fluid will enter inside the cell, forming pinocytic vesicle. Pinocytosis, the cell drinking fluid droplets enter the cell and forming pinocytic vesicle. What's the meaning of receptor-mediated endocytosis? Here is the cell membrane, okay? And I'm having receptors, which I told you present on the outer surface of the cell. And this is the ligand I want to enter. First, when the ligands come in contact with the cell membrane, the receptors will aggregate, okay, forming a pit. This pit from the inside of a cytoplasm will be coated with a protein known as clathrin. Okay, I'm going to let this ligand to enter the cell. Here is the cell membrane, here is the clathrin which is present here, and here is the receptor, and this is the ligand that enter. Specific molecules as drugs and hormones bind to a receptor that accumulate on the cell membrane. Gradual invagination of a cell membrane by the coated pit that surrounds the pit from the cytoplasmic aspect by a protein known as clathrin. Clathrin change the shape of the cell membrane from a pit and form a vesicle, and this vesicle will be present inside the cell. Okay, one MCQ question. The cytoplasmic side of a coated vesicle is surrounded by protein called what? Called clathrin. This is a very nice picture. Here is the cell membrane, and here is the clefran, and this is the ligand. The green is the receptor. The clefran will change the vesicle will change the bed into a vesicle. So this is the cell membrane containing the receptor and the ligand, and from outside is the clefran. When this coated vesicle come in contact with lysosome, the clefran will leave the vesicle, and the fusion will lead to moving out the contact between the receptor and the ligand, and the receptor will go back to the surface of the cell again. Exocytosis. After finishing the endocytosis, whether I do digestion or some sort of it, I'm going to have something undigestible. I want to get rid of it. Okay, so it will move by exocytosis. What the meaning by membrane trafficking? The including membrane movement and recycling developing in endocytosis and exocytosis. I have a part of a cell membrane enter during endocytosis. This part will regain to the cell by exocytosis. Okay. The first function of a cell membrane is transport. We have three types of transports, which is the passive, active, and bulk transport. Then we have the function of the surface coat, which is the glycolipid and the glycoprotein or the glycocalyx. Okay, first it contains receptors, so it's important for selective permeability. The other function is cell immunity, as it binds antigens to the cell surface. Cell recognition, it recognizes the cells of its own type, and this is very important during tissue rejection. Cell adhesion, the cell coat hold each other and to the extracellular matrix by the cell coat. Cell protection, meaning mechanical protection, protect the cell against the injury. Does all cell membrane have the same appearance? No. There are modifications in the cell membrane. First modification is called microvilli, and this microvilli increases the surface area, as in the cells, absorptive cells. Example, cilia push particles in one direction. For example, the cells lining the fallopian tube pushing the ova from the ovary to the uterus. Flagella, which is the tail of the sperm that allows sperm movement. Stereocilia, they are long microvilli present in organ known as the epididymis, and this is important for absorption. The last function is conduction of excitatory waves. The cell membrane, for example, of the lining the covering the cell, the nerve cell, it should receive nerve impulse. This nerve impulse will be transmitted on the surface of the cell. These are the cilia present on the apical surface of the cell that allow movement of structure in one direction. This is the stereocilia present in the epididymis in the male, and these are long microvilli for absorption. Again, this is the microvilli. These are the microvilli. They are increasing the surface area in absorptive cells. Clinical application. Non-functioning growth hormone receptor at the target cell lead a type of dwarfism. The level of the growth hormone in the blood is normal. If the receptors are not functioning well and the hormone level is normal, so this patient should be treated by something enhancing the receptor, not giving him the hormone, because the level of the hormone is normal. The hormone is not functioning to the receptor. And you should differentiate between the two conditions. I'm having a patient suffering from dwarfism. First, I'm going to assess the level of growth hormone. If the growth hormone is normal, so the abnormality in the receptor.